But with this auger, when we do it for antibiotics, what you want to get is a full lawn of bacterial growth. So there's 2 plates that you've got for this, and I did give everybody 2 plates, right?
No.
Okay, I'll pass out a second plate after I'm done with this. What you'll be doing is, one, you're going to use a throat culture because that's a very common thing to check for strep throat in there. When you do this, you're going to collect from the back of the throat. The other one, you're going to use either one of these cultures that you had to start out with. It doesn't matter which one. By creating a full lawn on here, that way you'll be able to see if there's any areas where growth is inhibited. So I am going to borrow someone's throat. When you do this, the trick is that you need to get back far enough because strep does grow in the back of the throat, but you're not looking to elicit a gag reflex in there. And by doing it fairly quickly in there, you don't have to worry about them usually gagging in here. If you make somebody gag and they vomit, guess who gets to clean it up?
The gagger.
Not me. It's going to be on your stuff. So So when you do this, what you're going to do is you're going to go over the entire surface. The idea is you want every single surface of this auger to have growth on it. Then you turn it and cover it again. Turn it a little bit. Do it again. Turn it again. And do that. So you've rubbed this swab all over the place. Because it does have human fluids on here, it's going to go into the biohazard. Thank you. And what we have are 3 different antibiotics we're going to test here. And so with each of these, you're going to use the tongs that you're going to sterilize. This is the one where you want to make sure you don't turn it upside down. These hold a lot of alcohol on it. If you go like this, it's going to run down and burn you. So you'll take 1 disc, and you want to put it about 1/3 of the way out on there. Just tap it down onto the auger. These are already marked. What these discs have in them is a known concentration of the antibiotic. So you want to have these spread out throughout the plate so that each one has its own area, that it can have a circle of growth inhibited on there. If they're right next to each other, you wouldn't necessarily know. So the more the antibiotic diffuses through the auger, if it's sensitive, it's going to stop growth. If it's resistant to it, it will grow right up to the edge of this on there. So when you look at it, it's going to be a pretty clear distinction of whether or not that organism is sensitive in there. So you can test multiple different antibiotics. I happen to have 3 of them in here, but there's a whole list of these that you could order. These, it's usually about $100 for a package of these. So anything in a lab is extremely expensive in there. And if for some reason they did not test a culture, it's just because of the cost of it, but it's really pretty cheap overall to test a culture to make sure that you get the patient on the right antibiotic and that you're not contributing to antibiotic resistance. So you'll have 2 of these plates, you'll bring them over, we incubate them, and you look at them next time. Any questions?
We want to mark and divide those into thirds?
You don't have to divide it into thirds because it's going to be the same organism over the whole thing.
And there's, it's identified what tabs there are.
Yeah, and these have the abbreviation on there for what exactly is the antibiotic in there. So I only have one container of each of these, so you'll need to share with these. Take one, use it, and then just bring it back so that the next group can use it on there.